Hello, welcome back to part 3 of our hormone lecture. In our third part, want to continue with the idea of insects producing hormones. And one of them that they're really good at producing is this hormone that's released from their body called a pheromone. Now, these chemicals, which act just like hormones, can serve as ways to mark trails, show territories, warnings, and so any hormone that's released outside of the body, like an exocrine, is going to be called a pheromone. And so in this picture you have a line of ants. You've seen the line of ants, and what they're doing is using their antennas to sense the concentration of the pheromones. And as they're walking, they're also releasing pheromones, thus strengthening the line, so they know exactly which way is to the nest and which way is to the food. Now, in this case, I want to focus on an experiment that was just released on a group of bees known as the undertaker bees. So the bee you see coming right at you carrying what looks like another bee is called an undertaker. So the undertaker is a worker, it's a female, and the one she is dragging that's right here in the front is a dead bee that she has just taken out of the hive. So the question really is, why would a bee take one of the dead bees out? So this is a research. Basically, ants, bees, and other insects are covered with these compounds called cuticular hydrocarbons, or CHCs. These are listed right here as these long alkane carbon chains. So these are all the different structures from different arthropods, specifically bees, ants, and wasps. And this gives this waxy coating on the outside that prevents them from drying out. And in doing so, it helps the insects also determine who's part of the hive and who isn't. Now, while the insects are alive, these molecules are continually released into the air and are used to help recognize members of the same hive. So how does an undertaker bee find a dead individual before they start to decompose in the hive? So this researcher did this question. He wanted to know if the amount of pheromones produced by the dead bee, or the lack thereof, or was it the body temperature, was the signal that the undertaker bees used. So their claim in the research was the amount of the CHCs produced by the dead bee, or their lack of, is the real reason that the undertaker bees are attracted. And the reason, of course, is pretty straightforward. A dead bee left in the hive too long would begin to decompose, and a fungal or bacterial infection could spread through the colony. So in their research, here's what the scientists did. They took dead bees and they warmed up a group of dead bees to about 90 degrees. The hive is about 95 degrees. They placed them back inside the hive. One was at 75, the other dead bee was at 90. And then they timed how long it would take before the undertaker bee removed him. For the one that was about 75 degrees, it took about 30 minutes, which is about average for an undertaker bee to find. But it took about 3 to 4 hours before the undertaker bees found the one that was warmed up. So the idea was maybe temperature was the reason. In the second experiment, they washed the CHCs off the bees themselves. So now the bees that were placed had no cuticular hydrocarbons on their bodies whatsoever. They repeated the experiment again with the same two temperatures, and the results were quite different. So I'm going to pause here for just a second and let you think about which experimental design supports this claim. So go ahead and pause the video. Go ahead and go back a slide or two if you need to, and think about which one best supports the claim that it was temperature or not temperature. In our last segment of this lecture, I want to kind of focus on 3 more parts about the hormones. The first, of course, is the pathways, like which way and how do the endocrine system works. And also, the second one is feedback. So I want you to know the difference between a negative and a positive feedback loops in the hormonal system. And finally, what is the difference between the steroid and an amine or a peptide hormone and how they work? All right, so let's kind of work our way through the pathways. So the first pathway is the very simple endocrine pathway. You have your stimulus, which then receives to the receptor. You have a signal transduction and then a response. This would be the release of the endocrine molecules. They enter the blood, go to some target, and then there's a response. So there's like a bigger stimulus, signal transduction and response, and then within the cell there's a signal, a transduction, and a response. So this is the simple pathway. Our next one is the neurohormone. So now we have the stimulus to the hypothalamus, to the posterior pituitary, which releases hormones. The hormones then travel to the target, and then there's a release or a response. And finally, a more complex pathway, which is the simple neuroendocrine pathway. So now we're going to do two steps. We have a neurological process with the sensory, a hypothalamus, a neurosecretory cell, release of one type of hormone that goes to an endocrine cell, which then releases a different kind of endocrine molecules into the blood, to the target, and then to the response. So those are the three pathways. They are the simple neuroendocrine, the neurohormone, and the endocrine itself. Those are the three pathways. Let's look at the feedbacks. A negative feedback, the response produced by the target cells, would then go back and prevent the stimulus from targeting the endocrine cell in the first place. So in this example is how we secrete something called secretin, which is part of the digestive acids. Here you have secretin released by the pancreatic cells into the stomach and into the small intestines. It releases bicarbonate, which are going to then neutralize the pH from the stomach. And so this process, once you have enough bicarbonate and the pH returns to neutral, that's going to feed back and tell the pancreas to stop producing secretin, which would then tell the pancreatic cells not to produce the bicarbonate. A positive feedback, not many exist in the endocrine system, but this is one of them. The nursing of mammals causes a sensory input to the hypothalamus to release something called oxytocin. Oxytocin targets the mammary glands. The mammary glands produce milk. So as long as the animal is nursing, then the animal is going to produce milk, and that's a positive feedback. So the more there's milk production that comes back, causes more nursing produces oxytocin, which causes more milk to be produced, and that's a positive loopback system. Now, the three types of peptides, amines, and steroids, these are the different types of hormones, specifically in vertebrate animals. And let's see how they move through this signal transaction. There's a reception, signal transduction, and response. And let's see how it works.
Hormones act as chemical signals, but how do they trigger changes in particular target cells? Non-steroid hormones, hormones derived from amino acids, oxytocin and epinephrine for example, work via signal transduction pathways to activate or inhibit proteins within cells. These hormones can trigger an immediate response in target cells by turning cell components on or off. A non-steroid hormone binds to a receptor protein in the plasma membrane. The receptor activates a signal transduction pathway in the cell. A series of relay molecules transmits the signal to a protein that carries out the cell's response. In this case, an enzyme is activated. A different non-steroid hormone might trigger a different response, perhaps turning on specific genes that result in the synthesis of a particular protein.
All right, now let's see a steroid hormone.
steroid hormone, such as testosterone or an estrogen, acts by entering a target cell and turning genes on or off. Steroids usually act rather slowly, because it takes time for the gene products to build up or become depleted. This steroid hormone passes through the plasma membrane and binds to a receptor protein in the cytoplasm. The hormone receptor complex enters the nucleus and attaches to specific regulatory sites on the target cell's DNA. There it acts as a transcription factor, a gene activator. A messenger RNA molecule is transcribed, which is then translated into a protein, an enzyme, for example. This new protein alters the function of the target cell.
So this is going to go ahead and stop our part 3. In our last part, I will give you some specific examples that will lead you into your activity for this week.